Bismillahirrahmanirrahim. Welcome back to my channel. In this video, I'll discuss the types of membrane proteins and their functions. The cell membrane is best described by using a structural model called the Fluid-Mosaic model. According to this model, the molecular arrangement of the plasma membrane resembles a continually moving sea of fluid lipids that contains a mosaic of many different proteins. Membrane proteins are classified as integral or peripheral according to whether they are firmly embedded in the membrane. Integral proteins extend into or through the lipid bilayer and are firmly embedded in it. Most integral proteins are transmembrane proteins, which means that they span the entire lipid bilayer and protrude into both the cytosol and extracellular fluid. Like membrane lipids, integral membrane proteins are amphipathic. Their hydrophilic regions protrude into either the watery extracellular fluid or the cytosol, and their hydrophobic regions extend among the fatty acid tails. Some integral proteins form ion channels or poles, also known as holes, which allow flow of specific ions such as potassium to get into or out of the cell. Most ion channels are selective, that they allow only a single type of ion to pass through. Other integral proteins act as carriers, selectively moving a polar substance or ion from one side of the membrane to the other. Carriers are also known as transporters. Integral proteins called receptors serve as cellular recognition sites. Each type of receptor recognizes and binds a specific type of molecule. For instance, insulin receptors bind the hormone insulin. A specific molecule that binds to a receptor is called a ligand of that receptor. Some integral proteins are enzymes that catalyze specific chemical reactions at the inside or outside surface of the cell. Integral proteins often serve as cell identity markers. They may enable a cell to either recognize other cells of the same kind during tissue formation, or they may recognize and respond to potentially dangerous foreign cells. The ABO blood type markers are one example of cell identity markers. Peripheral proteins are not as firmly embedded in the membrane. They are attached to the polar heads of the membrane lipids or to the integral proteins at the inner or outer surface of the membrane. Some peripheral proteins on the surface of intestinal cell, for example, act as digestive enzymes to break down nutrients to sizes that can pass through the cells and into the bloodstream. They also act as identity markers. Peripheral proteins help support the plasma membrane and participate in changing cell shape during cell division and in muscle cells, and attaching cells to one another. Many integral proteins are glycoproteins, proteins with carbohydrate groups attached to the ends that protrude into the extracellular fluid. The carbohydrate portions of glycolipids and glycoproteins form an extensive sugary coat on the membrane called the glycocalyx. The pattern of carbohydrates in the glycocalyx varies from one cell to another. Therefore, the glycocalyx acts as a molecular signature that enables cells to recognize one another. For example, a white blood cell's ability to detect a foreign glycocalyx is one basis of the immune response that helps us destroy invading organisms. In addition, the glycocalyx enables cells to adhere to one another in some tissues and protect cells from being digested by enzymes in the extracellular fluid. The hydrophilic properties of glycocalyx attract a film of fluid to the surface of many cells. This action makes red blood cells slippery as they flow through narrow blood vessels and protects cells that line the airways and digestive canal from drying out. Thanks for watching and listening. Please do share this video with your friends and colleagues.